Vamos a comenzar la segunda jornada del encuentro sobre el síndrome de Rett. En el día de ayer recordarán ustedes cómo abordamos los aspectos más generales en materia de investigación y también tuvimos una mesa redonda con pacientes, con familiares de niñas con síndrome de Rett. Y hoy entramos ya en una materia mucho más específica y una materia que seguramente pues ya a ustedes ya pues les genere mucha más ilusión. Y digo que la materia de hoy ya puede generarles mucha más ilusión porque lógicamente ya vamos a contar con una serie de investigadores de gran altura que ya nos van a hablar de una manera muy específica del gen MPC2 y también pues nos van a informar de las líneas de investigación que están siguiendo, algunas de las cuales pues la verdad es que son muy prometedoras. Yo creo que va a ser una jornada muy interesante, por eso pues yo no voy a hablar mucho más. El primer ponente de la mañana va a ser Stuart Cobb. Stuart Cobb desarrolla su trabajo en el laboratorio de la Universidad de Glasgow. Es un experto en circuitos cerebrales, en salud y enfermedad, y también en abordajes farmacológicos y genéticos en los trastornos del sistema nervioso central. Su contribución particular en este encuentro es la de actualizar para todos nosotros el trabajo que está desarrollando referente a la terapia génica, algo de lo que se habló ayer, la terapia génica aplicada al síndrome de Rett. Ha habido progresos significativos recientemente y Stuart Cobb nos los va a comentar. El título de su ponencia es Terapias para el síndrome de Rett dirigidas al MECP2 a nivel de gen y proteína. Stuart, cuando quieras.
Better? Yeah. Okay. I'll start again. Okay, so good morning. I'd like to thank the organizers for inviting me today. And I guess this starts the scientific session. So what I'm going to talk about today is a bit of an update on our work looking at therapeutic approaches in Rett syndrome, and particularly those targeting the gene and the protein MECP2. Now, this first slide, this was shown by Monica yesterday. So these are some of the priorities for research where the most resources are being invested. So there are essentially two main types of therapeutic approach. One is to treat the disorder, treat the symptoms of the disorder, and a lot of these are repurposing existing drugs and looking at small molecules. And then there's a bit to the left, cure there on the graph, and that's really targeting the root cause of Rett syndrome, so targeting the faulty gene, either by replacing the gene with a healthy copy, Rett syndrome, gene therapy, or by fixing the mutations at the mRNA level, or through replacing the protein rather than the gene. So there are people working at all these different levels. So for gene therapy, there are, Monica mentioned one company yesterday. There are actually other companies working on gene therapy for Rett syndrome as well. And for the protein therapy, there are two companies currently working on protein therapy for Rett syndrome. So there's a lot of activity that you don't hear about that's going on within industry as well. So the first question I'd like to address is what aspects of Rett syndrome might we be able to treat? So this is really a fundamental question. So what features of Rett syndrome are fundamentally treatable? So to address this question, so this work goes back 10 years from the original paper with Adrian Bird, showing that in mice, if you switch off MECP2 in mice, mice develop many of the features of Rett syndrome. So they have movement effects, there are effects on breathing and autonomic dysfunction. So there's a whole range of features that are reminiscent of Rett syndrome. And what you can do using a genetic trick is you can switch the gene back on, and you can ask the question, well, what features, Rett-like features, improve when you switch the gene back on? And to summarize a whole number of studies, the bottom line is that most things we've looked at, whether it's motor activity, in other words, movement activity, or whether it's abnormal EEG, or whether it's breathing, abnormal breathing, most of the things that have been looked at do improve if you switch the gene back on. So there's actually good evidence, at least in mice, that if you can restore the gene, then you would be able to produce a reasonably profound improvement in Rett syndrome. So that's the first question. So that's encouraging. So the next question is Rett syndrome purely a brain disorder? Because MECP2 is actually, the protein is actually found throughout the body. So there's no cell in the body, at least mature cell, that doesn't have MECP2. It's expressed everywhere. So what we asked here is how much of Rett syndrome is really due to problems of the nervous system? So what we've done is we've got a mouse, normal mouse, and then on the right we have a mouse in which the MECP2 gene has been switched off. And then in the middle there's a mouse in which the MECP2 is switched off everywhere other than the brain and nervous system. And we asked the question, you know, what is the difference between these three mice? And the bottom line is when you switch off MECP2 in the periphery, everywhere other than the nervous system, you have quite a normal mouse. So it's really saying that the nervous system is at the center of this disorder, and that's the main tissue to target for therapies. So there are a few minor features that you can detect which are genuinely due to lack of MECP2 in the periphery, but they're very minor. So the major features, so you can see in terms of behavior, abnormal breathing and so on, they reside in the brain, dysfunction in the brain. So the brain is the major target for these therapies. So the next thing, MECP2 protein, the levels of protein varies a lot from cell to cell. And so it tends to be in peripheral tissues, it's at quite low levels. In the brain, it's at much higher levels. But even in the brain, from cell type to cell type, the levels of MECP2 vary quite a lot. And in fact, the level of MECP2 is probably going to be very important when thinking about therapies, because you need to get the levels right. If you have too little MECP2, functional MECP2, you have Rett syndrome. If you have too much MECP2, well, there's a duplication syndrome. So the correct level of MECP2 is really critical. And this is actually a big challenge for therapies targeting the gene and the protein, getting the levels right. That's the most difficult part. Because you can see that the cartoon on the right shows that all girls with Rett syndrome are mosaic. That is, they have some cells that have MECP2 present at the normal levels, and then other cells have either faulty protein or no protein. And it's really those cells that you want to target without producing too much MECP2 in the cells that already have it at the right level. So that's a big challenge. Okay, so why gene therapy? So some of the speakers touched on this yesterday. So the reason that gene therapy holds promise in Rett syndrome is that Rett syndrome is a single gene disorder. So MECP2 is a relatively simple gene, and it's a simple gene which, when mutated, and you get a loss of function, results in Rett syndrome. And the big advantage of targeting the gene or the protein is that you target the root cause of the disease. So conceptually, any therapy would be what's called disease modifying. So it's not treating the symptoms, but it's actually treating the underlying cause of the disorder. And so this means that, in fact, we don't need to know a lot about what MECP2 actually does. So there's still a lot of debate as to what MECP2 actually does in the brain, and I'll touch on this later. So you don't need to know that for gene therapy. All you need to know is you just put the gene back. And then lastly there, MECP2 has an ongoing role. So, Rett syndrome, if you think of it as a neurodevelopmental disorder, so MECP2 is not just essential during development, it's essential throughout life. So it has an ongoing role in the nervous system. It's not essential during a critical period. If you take a mouse, for instance, and switch off MECP2 as an adult, it will develop breathing disturbances, it will develop motor effects. So MECP2 is having an active role. So replacing the gene or protein at any developmental stage, including the adult, should have a beneficial effect. Okay, so this work began a few years ago. So the concept of gene therapy is actually quite simple. So you take the gene of interest, MECP2, and you package it in a virus, and then the virus is in a solution, and then you deliver that virus by injection. And so we tested this a number of years ago in mice that model Rett syndrome. And what we observed, so these are male mice, so a lot of the studies in mice use male mice rather than females because they, like in the human disorder, they're much more severe, and therefore any effects you see more quickly and are more pronounced. So in these mice, what we observed with the gene therapy is that when you deliver this virus, delivering MECP2, just like any drug, at low doses, we see no effect. As you increase the dose, we start seeing a therapeutic effect. So this is just, so the males are very badly affected, so this is actually just survival of the males. So you'll see that you get an enhancement of survival, you get a reduction in the neurological score, if you like. But as you increase the dose of the virus, it starts having a detrimental effect. You start delivering too much MECP2, and this is deleterious. And actually, one of the big problems is when you deliver the virus, it accumulates in peripheral tissues at very high levels. So the slides on the bottom right there shows the liver, and any drug tends to accumulate in the liver, and if you deliver too much MECP2 to the liver, that's toxic to the liver. So normally MECP2 is very low in the liver. So what we did is we went on to modify the gene therapy cassette, as it's called. And what we did is we built in regulation to regulate the levels of the gene. So we adjusted what's called the promoter, which is the switch that turns on the gene and sets what level it's expressed at. And we also adjusted what's called the 3' UTR, and we built in some of the normal regulatory sites that are in the gene, which turns it down in certain tissues. And once you do that, you have a much better gene therapy molecule that is much more tolerable. It doesn't cause the liver toxicity. It doesn't cause neuronal toxicity because it's more tightly regulated. So if you now, we call this a second generation vector. So if you now deliver this to mice, so again, these are male mice, you'll see in the bottom left, this is their survival. So normally the male mice, they don't survive. So half of the mice are dead by 11 to 12 weeks. But you can see with the virus-treated ones, approximately half the mice, or just under half the mice, are still alive at 1 year. So it's producing quite a profound effect. And in the third graph along, sorry, I don't have a pointer, where it says score, this is neurological score. So if you like, this is a score for the neurological features in the mice, and what you see is the black dots, that's the score of the untreated mice, and the red is the treated. So you're getting an improvement or a lowering of the neurological score. Is that the pointer? I think the trouble is the screen, it doesn't show up on the black screen. It does something weird with lasers. Okay, so this is very encouraging. So this is the second generation vector, and in fact, this is just showing data from one, but we've got quite a range of these. We've got lots of different cassette designs that we've been testing to see which is the most effective. Now, people with Rett syndrome, a lot of Rett syndrome mutations actually, there are still MECP2 protein present, but it's non-functional protein. And one of the concerns with gene therapy was that if you were delivering a gene and there was already the faulty gene there, they might interfere with one another, and this may limit the effectiveness of the gene therapy. But this appears not to be the case. So this is now a different mouse model. This is what's called T158M. So this is the most common mutation that causes Rett syndrome. And you can see in these mice as well, you get a very clear survival effect. And again, the red dots shows the therapeutic vector. So it's causing a big drop in the neurological symptoms when it's delivered to this mice. So this is encouraging both in the knockout and in mice that more accurately model Rett syndrome. So this is an actual Rett syndrome mutation. So as Monica mentioned yesterday, so one of the lead vectors has been taken forward by Avexis, which is a company headquartered in Chicago, to develop a clinical gene therapy program in Rett syndrome. So that's quite exciting. So this is based on mouse data and also data in non-human primates, safety data. They plan to go ahead, hopefully with a clinical trial, if they get approval. Okay, so what we're doing is actually, there's lots of variables for designing the gene therapy cassette, and this is our work moving forward, is trying to make better and better vectors, next generation vectors that will be even more effective. And there's lots of things that you can modify. So you can modify the promoter, you can modify the gene itself, or the protein itself, and you can modulate the regulation of the gene, and also the type of virus that you put it in, because the technology's evolving all the time. It's developing very rapidly, so we're keeping up with that and trying to develop better and better vectors. Now, the next few slides are more for the scientists, so this is really to tackling what is MECP2 actually doing in the brain or in neurons at the molecular level. So MECP2 is a nuclear protein. It's found in the center of the cells, as you can see in the image there of a neuron. And essentially, MECP2, there's lots of theories of what MECP2 actually does. So it seems to regulate gene expression or processing in a number of ways. But one of the key features is that it has a binding domain. So it binds to DNA, and it binds to methylated DNA in a particular context, and it also binds to other proteins. So it forms a bridge. And if you look at the mutations that cause Rett syndrome, so these are point mutations that just change a single amino acid. What you find is that they tend to cluster in these two bits of the protein that cause binding either to the DNA or to this, what's called a repressor complex. So what we asked is, well, do you need the other bits of the gene at all? So what we did is we made mice. So this is Becky Tolliston's work in Adrian Bird's lab. What we did is we made mice in which we cut off the left-hand bit, so the grey bit on the left, we cut off. We then cut off the bit on the right as well, what's called the C-terminus. And then we decided, well, let's go the whole hog, let's cut out the middle bit as well, and just keep these two domains, this methyl binding domain and this repressor domain, in this mini protein. So this gene is now a third, it's actually less than a third of the full-length gene. And ask, well, what happens? And very surprisingly, in mice at least, very little. The mice are actually quite normal. So if you cut off the left-hand end, the N-terminus, or the left and the right-hand end, the N and the C, half the length of the gene, the mice are indistinguishable from wild type. They appear completely normal. And when you cut out the middle bit, so this delta NIC, there's very subtle phenotypes. So you can see the severity score. So the null mice, they get more severe and then they die. But you can see that this protein, which is a third of the full-length protein, it actually has very low neurological score. So very, very subtle phenotypes. So the question is, well, what's the significance of this? Well, there are two. One is scientific. So one is that MECP2 has said to do lots of things. So there are lots of bits of MECP2 that people have said are important. So it involves things like lots of phosphorylation sites, AT hooks, binding to different parts within the cell or different processes. So these mini constructs have none of these. They only have these two domains. So it really suggests that, so I'm not saying that these other things don't actually do something within the cell. All we're saying is that they're not fundamental to Rett syndrome. So there's a scientific element there, but also in terms of therapy, because if it turns out that we can just deliver a small fragment of MECP2, it opens it up. It means it's much more deliverable. So MECP2 is quite big. So to get it in a virus, it's quite challenging to get that regulated. If you want to deliver it as a protein, it's quite a big protein to deliver. But if you can deliver a truncated version, it becomes much better therapeutic molecule. And that's what we're doing moving forward. So this is a gene therapy experiment again, but this time using this small fragment of MECP2. So you can see the graph on the left. The pink line has come down. So that's the improvement in neurological score based on having just this small truncated element of MECP2. And we've done some behavioral phenotyping to show that there's an effect as well. So really our work going forward is working on this plus the full length. And because it's much smaller, it allows you to do a number of other things. So one thing that Monica mentioned yesterday is that there's inherent problems of gene therapy in terms of safety. So unlike a normal therapy where you deliver a medicine and if it has a therapeutic effect, great, but if you have adverse effects or negative effects or it's not working, you can withdraw treatment. With gene therapy at the moment, you can't do that. It's a one-off delivery and then that's it. There's no going back. So we're looking at approaches where you can actually switch on and off MECP2 once you've delivered it. So we're looking at what's called tunable systems. So these are either promoters, these switches that you can switch on and off pharmacologically, or making forms of the protein that you can regulate pharmacologically. So so this shows an example of one. So this is so the green dots is MECP2 in cells, and you can see that in the second figure along, you can see that there's very little green dots. And this is because this MECP2 has been modified so that it's degraded rapidly. But when you give a drug which crosses the blood-brain barrier and gets into the brain, by increasing the concentration of that drug or dose of that drug, you can increase the level of the MECP2. So this is an example of the type of system that you might have where you can actually, you deliver your gene therapy, but then you can regulate the level of MECP2 dynamically after it's been delivered. So this is just examples of ongoing work. So we're developing better gene therapy cassettes. We're trying new viruses, new vectors that are being developed all the time. We're looking at tunable systems, and then we're looking at approaches where you're not delivering just the whole MECP2 gene, but you're trying to just reintroduce small elements of the gene which may have other advantages as well in terms of controlling levels. So I'm sure I'm over my time, so I'll just round off there. So I think there's a number of factors that support the concept of gene therapy. So it's not reached clinical trial yet, but there's a lot of encouraging data to suggest that it's an exciting way ahead. We know that when you deliver MECP2 to the nervous system, you can improve a lot of the features of Rett syndrome. So most things that have been looked at seem to improve. There's a problem with delivery, so we think probably direct delivery to the brain is going to be essential for delivering MECP2, because when you deliver it to peripheral tissues, that may be problematic. So the challenges ahead are shown here. So one is we need to target a lot of cells across the brain to get a profound improvement. And the other one is then achieving the correct levels of MECP2 in the appropriate cells. And that's really the bulk of our work moving forward. So in the last slide, just make some acknowledgements. So people in my own lab, and then a lot of the mouse works in collaboration with Adrian Bird's group, and then the gene therapy work is a collaboration with Steve Gray and Brian Casper and Gail Mandel at various gene therapy centers in the United States. So I think I'll stop there. Thank you for your attention.